Hello and welcome to another video lecture from MrWatkins.com. This one is about the electron transport chain. You should have already seen or at least know about the Krebs cycle and how that process works before you watch this particular video. Please remember that in the Krebs cycle there were some molecules that were made. So let's just go back over those real quick. We had carbon dioxide made. Carbon dioxide is a gas that is given off, and we had 3 of those made in the process of one pyruvate molecule coming through. We had ATP formed. So ATP storing energy, we just had 1 of those molecules for the pyruvate that went through the Krebs cycle. We had NADH, the reduction of NAD to NADH, storing energy. We had 4 of those occur in the Krebs cycle. And then we had another electron carrier, FADH2, and we had 1 of those formed. Remember that CO2 gas given off, so that's going away. ATP, what we want, but it's these 2 molecules that we're going to focus on as they go through the electron transport chain. So when we look here on our flowchart that you should have already downloaded, each NADH that's made, and we have 4 for every pyruvate molecule, is going to start the process of the electron transport chain. Now, because this is pre-AP, there's a lot of things that are missing here that you're going to pick up with the AP or the IB biology class that you may take later. But needless to say, when NADH starts this process, it's going to form 1, 2, 3 ATP. Now this will only happen, and think of it almost as a stair step, as the energy kind of works its way through this membrane. It will only happen if at the end of this process, oxygen is there to pick up some electrons, to pick up these hydrogens and form water. This is where the water is formed in the cellular respiration equation. So that each NADH now is going to create or be converted, the energy is going to be converted into 3 ATP. Now FADH2, it enters farther down the system, so it doesn't create 3 ATP, it only creates 2 ATP. And again, in this process, there's a lot more detail, but taking IB and AP bio, you'll get into that detail. Remember that this membrane here is the inner membrane of the mitochondria. Specifically, this is the cristae. Now, to fill this part out, to figure out how much is being made or converted, we need to make sure that we have all the NADHs. And if you remember back in glycolysis we had, in glycolysis 2, we had 2 NADHs that were formed right after the split. And those 2 NADHs get moved to the inside. So we had our 4 NADHs that were formed in the Krebs cycle, plus the 2 other NADHs, or excuse me, the 1 other NADH from the pyruvate being made, 1 pyruvate being made. That gives us a total of 5. But remember that with glucose, 2 pyruvate molecules are made. And so the cycle, the Krebs cycle, has to go around 2 times. So we're going to get 2 turns of the Krebs cycle. Put these molecules back where they belong. 2 turns of this Krebs cycle here. So that means 4, 8, and we're going to have 2 NADHs come through. So we're going to get a total of 10 NADHs being produced. A total of 10 NADHs, which gives us a total of 30 ATP being produced in the electron transport chain here on the end. With the FADH2, again, because this process of forming the 2 pyruvate molecules, we got 2 pyruvates coming through. Krebs cycle is going to turn twice. 1 FADH2 is being made. So that means we're going to have 2 total FADH2s per 1 glucose molecule. That's going to give us 4 ATP. So our total ATP made in this electron transport chain is roughly 34 ATP. If you take that into account, along with the 2 ATP being made in the Krebs cycle, so there's 34, 35, 36, plus the 4 being made in glycolysis, we're going to get a gross gain of ATP of 40 ATP molecules. We're going to have 40 total ATP molecules. And we're going to end up spending 2 ATP molecules here to get the NADHs into the mitochondria. And we also, if you'll remember glycolysis, we spent 2 other ATP to increase the energy in glucose to fructose 1,6-phosphate so that we could split it. So we're going to spend a total of 4 ATP. That's going to give us a net gain of 36 ATP. 40 all made, 34 of that 40 all made here in the Krebs cycle, and then the rest of it made throughout the rest of glycolysis and made off of the Krebs cycle. That's 36 ATP. That's a lot of ATP. So your total number of ATP produced is going to be 36. The energy required is 12 kilocalories per mole divided by 686. You're roughly going to get 70% efficiency here. You're getting a lot more ATP than you did when you're going to just use glycolysis. That's basically it. The next video you may want to watch is the one on anaerobic cellular respiration.